IB Environmental students, today we're going to be talking about the different methods of monitoring pollution. So some of the methods can be direct methods. Those are actually measuring the pollutant itself. This can be done by test strips or by probes. But if I'm measuring the nitrate levels, I'd actually measure the nitrate levels. If I want to measure the acidity, I'd measure the pH level and so forth. So direct measurements are measuring the actual pollutant. This is some examples: measuring temperature in several locations along a river at different times of year. That would be measuring the amount of thermal pollution. Measuring nitrate levels as an indication of nutrient overload. This could be done in water or soil. You would have to describe the method. So a method would be taking a baseline measurement and then monitoring systematically over time or in different locations to determine any changes. Indirect methods include a lot of different things, but overall an indirect method is monitoring the effects of a pollutant by checking other factors. This makes a lot more sense as we go over some examples. Some examples are measuring the dissolved oxygen or the biological oxygen demand. These will actually change as we have other pollutants in an area. So there are indirect ways of measuring how pollutants are causing damage. Another way is to measure the presence or absence of indicator species. We're going to get some examples of that in a little bit. All right, let's dig in. So first we have to understand what biological oxygen demand, or BOD, is. All right, this is one of the hardest things for students to understand in these notes, so make sure you take your time. It is the amount of dissolved oxygen used by aerobic or oxygen-using bacteria as they break down organic or dead matter in a specific volume of water. So the important thing is it is the amount of oxygen used by bacteria. So sometimes remembering B for bacteria is very helpful. So how much are they using up? This will occur more if there's more dead matter. So what do the results tell us about the amount of pollution, since this is an indirect method? Well, they'll tell us if there was a lot of organic matter around. This could be due to sewage or agricultural runoff or fertilizers. If there's a lot of organic matter, that's going to cause a lot of biological oxygen demand because the bacteria will have a feast on all the dead yucky stuff. All right, so we as scientists in the local environmental areas, they will label the amount of biological oxygen demand, and they will use it as an indicator of overall water quality, which would relate to other pollutants like sewage or nitrates, etc. All right, and they'd categorize it. Let's learn a little bit more about biological oxygen demand. So when we think about this, we actually have to be able to describe how it relates to dissolved oxygen, or DO, and how it impacts the health of a water area. As we look here on the chart, we can see that this orange line of dissolved oxygen actually decreases as the amount of biological oxygen demand increases. The septic zone is the zone where there's not a lot of things living. That's when BOD is at its highest, the blue one. It's kind of high in that area, and dissolved oxygen is at its lowest. That is when we have a lot of dead things that we're decomposing for the bacteria to have a feast on. That's our biological oxygen demand. Hopefully over time things would recover and we'd go back to normal, but this is our area we're going to talk about the most over the next couple weeks. So to summarize that, if my dissolved oxygen is greater than my biological oxygen demand, my system is considered healthy. I want more dissolved oxygen than the amount of oxygen taken up by bacteria. If my dissolved oxygen is less than my bacterial demand on the oxygen, then it's possible I don't have enough oxygen to sustain large organisms like fish. It is quite a challenge to learn how they actually measure biological oxygen demand, so that will be something we'll do in class if we have time. Now for some other indicators or biotic indexes that are also indirect measurements of different types of pollutants. They don't measure the pollutant themselves. So this is measuring if an organism is present All right, an indicator or biotic index are species. If they're abundant or absent, they indicate if a pollutant was there. There are lots of different types of species that are very sensitive to pollutants, and they are really good indicators that pollutants are present. There's also ways of doing an overall catch of the species, comparing to normal and later after a pollutant, and seeing if the diversity index has changed. We know that a diversity index is a specific name that we've learned. What was it called? It's called Simpson's diversity index. It was that weird thing with the sigma. How do we calculate it? I would go back and look at Simpson index. I'm not going to revisit it a ton right now. But we do need to understand the overall difference between abundance and evenness. We really, for a healthy, stable system, we want a lot of evenness because we want a lot of different types of species. So to help us, we're going to kind of go through some examples of some indicators that would show the presence or absence of a pollutant. So our big one would be macroinvertebrates. A lot of local environmental groups go out to streams like Akatank or the Potomac, and they will measure the amount of macroinvertebrates at the beginning of a time of year and then later to see if there's any changes. What they do is they catch these very small bug larvae, all right, such as stoneflies, mayflies, caddisflies, tons of little diverse bugs. And if there are a lot of them, that means it's a very healthy area, and they can do a diversity index on that. We could also collect different times of year the amount of coliform bacteria. That indicates the presence of sewage waste, which tells us, is our wastewater treatment area leaking? Is it working? Overall, all of these things tell us about diversity. We have to do methods over time of getting multiple samples, comparing to baselines, comparing to legal limits. And those tell us indirectly if there's a pollutant and give us some indication of what we might want to do moving forward. Good job, guys!